You're listening to the Interactome, a podcast by a group of young researchers who want to connect you to the world of science by sharing their stories and perspectives. Just in case their bosses are listening, they want to remind you that the opinions expressed here are their own. They also want to remind you not to take anything they say as medical or professional advice, as they are not doctors. Not yet, anyway. Stay tuned about that. And, without further ado, welcome to the Interactome.
Hi everyone, welcome back to the Interactome. If you've forgotten who I am by now, because I haven't been on a couple episodes, I'm Sam. I am a biochemist, but that's actually not really important for this meeting, because I'm actually going to disappear. But I'm here to introduce our guest today, who I know very well. Yeah, so our guest today is actually my girlfriend, Margaret, who...
Woo!
Hi guys.
Who is a scientist in her own right, probably more of a scientist than I am at this point. And she studies something called mass spectrometry. So she'll go into explaining what that is, because obviously she knows more about it than I do. But she's joining us today because she does some really cool science and she works with some really cool people. So I'll let her and everyone else in this episode, so that's Joe, Natalie, and Sarah, kind of take control while I go and be quiet in the other corner, building a little model piece of heavy equipment, which is, I guess, a new hobby I picked up because I clearly have some opinions already about everything she's going to say. I've heard half of these stories, and so I'll be like, wait, that's what you were talking about? So I'll let you all go, but I just wanted to introduce our guest because, you know, I dragged her here.
Well, I'm happy to be here.
Yeah, Sam, thank you so much for that introduction. And listeners, welcome back. I'm Natalie, science communicator here at the Interactome, 1/3 of our hosts this week. And so I'll turn it over to my other co-host for this episode to just give a little introduction.
Hey everyone, I'm Joe, currently a med student at the University of Pennsylvania, but also hoping to do a PhD as well. Big science nerd, very into molecular biology, and that's kind of my thing. But I'm very excited to learn a bit more about kind of like the chemistry, sort of physics kind of side of things, and I'm looking forward to that.
Hey everyone, I'm Sarah. I agree with Joe. I'm also a medical student. I don't really run into a lot of chemistry and physics on the regular as much anymore, so I'm excited to hear more about what Margaret has to say. So I guess given that, Margaret, if you want to give us a little bit of background about yourself, where'd you go to school? What was your background like? How did you get to where you are today?
Sure. Thanks again for having me. I graduated from Northeastern University in 2019 with a degree in biochemistry. And during that time, I had sort of a mixed biology and biochemistry background. I worked in a bacteria lab as an undergrad, but what I also did at Northeastern were a couple of internships, and one of those was in a mass spec core lab. And that was sort of where I learned to appreciate the power of the technique and what it could do to help us understand the chemical basis for disease and for therapeutics. I'm a PhD student now. I mostly study the molecules in the brain, but very broadly speaking, I'm going to quote my PI here. He considers himself a biochemist with a capital C. So sort of with an emphasis on the chemistry, but wanting to focus on living systems and how they can go wrong.
I think that's so cool. And so you've mentioned that what you do is, you called it a technique, I believe. So we've brought up the name quite a few times, but I'm going to say what everyone's asking. What the heck is mass spec?
Oh, that's an episode. That's
Yes,
the episode
Natalie.
title right there.
That's the title.
That's the episode title right there.
Yeah, so mass spec is sort of the dictionary definition is it's an analytical technique that is used to measure the mass to charge ratio of ions. And a way that I heard it described in class that I actually really like is you can think of it like a scale that weighs really, really small things, like individual proteins or even parts of proteins. Yeah, that's sort of what I spend my time doing. And then what I can do is I can take a cell sample or a tissue sample, I can cut up the proteins into little pieces, and because that is what the mass spec is able to weigh. Like, you can think of a scale having a capacity. There are special mass specs that weigh really big things, really big in this case meaning whole proteins, which are of course still very small, but that is sort of the scale at which I work.
And what is the advantage, or what can those samples tell you, even if they're so small, if you're weighing them? Like, what sort of insights can those give you as a scientist?
So you can get information about how abundant a protein is in a sample.
Okay.
I spend a lot of my projects focus on neurodegenerative disorders, so I'm very interested in which proteins are more or less abundant in, let's say, a disease like Alzheimer's disease. And what I can also do is I can get very specific about the ways that they are changing. So proteins are dynamic. Proteins are not set in stone. They can have things stuck to them or removed or be cut in half or really any number of things. And the mass spec provides information about how that happens and when. When during the course of a disease might this be the case?
So if you are investigating a disease, in your case, you look at particular molecules in the brain, do you maybe wait for an event to happen, right, that could possibly trigger the proteins to fall apart, change shape? Or is it kind of like you're sampling kind of randomly to kind of pick through? Yeah, interested in your thoughts there.
Yeah, so I'm sampling pretty randomly.
Okay.
I get sort of a population of people who have passed away and donated their brains to this particular study. And then our neuropathologist colleagues can provide some diagnostic information, and then they can just send them to us and we can run our experiments. And I think what I should say too is, I alluded to this a little earlier, I can do this on sort of any type of sample. I can take sort of cultured cells, I can take tissues. I have a project right now that looks at arteries from people's brains, so I'm not always just looking at brain. And again, I am trying to say what and how much of each thing is in a particular sample.
Do the samples look like brains? Like when they come in, are you like, oh my gosh, that's a brain? That's cool.
I mean, the brains, when I use them, they tend to be frozen, and I tend to be making sections of them, so it's not quite so. It doesn't feel as much like a brain, but I tend the things. I do work with mouse brains pretty frequently too, just for method development. Mm-hmm. Because I want to. A mouse brain I can buy for cheap. A human brain is precious.
True.
And so I work a lot with mouse brains, and those are a little disturbingly squishy. We don't have to keep that in, but I was working with the arteries just sort of late last week, and those feel they're very organic.
Is it stressful to handle something so small, like when you're sampling? Because I also feel like if you're doing a technique that your samples are so itty bitty and you're measuring like mass to ion, like ratios to ions, can that be like stressful when you have like only a limited amount of sample and you have to get all this information from it?
Yes, it can be. So the sample is still visible to me before I go and start to process it. But once it is in solution, I can't see the proteins. I just have to sort of trust that they're there. And there's a lot of steps where they can go missing, so to speak. And so especially when I do an experiment for the first time, I save fractions. I save samples of each step so that later, if I run it on the mass spec and don't see anything, I can retrace my steps and say, okay, where did I lose my sample?
This is kind of a related question, but I just want to make sure I understand. So in general, the goal of mass spec is to weigh pieces of proteins. And by seeing how much of each protein we have in a sample, we can kind of get a sense of what's going on inside, like the brain tissue or the artery or things like that. And you can tell that the quantity of a protein is different in like your control versus your treatment because you have just more of one in the other. Yes. But how do you actually like differentiate between protein A and protein B in a sample? That seems like for me, like I know like in my experiments I have antibodies that will stick to one protein but not the other, and I can use that to determine which protein is which. But in your case, you're working at an even smaller scale, and so I'm just very curious about how you can, like, based on what I understand about mass spec thus far, it seems like you can get a sense of like just little, like the size of little pieces of proteins, but like how do you match that to an identity of a protein, if that makes sense?
No, this is a great question. And gets at a really key part of the experiment. So what we do, we essentially, after we run a mass spec, we end up, for lack of a better way to put it, we end up with sort of a list of mass to charge ratios that are observed. Those can then be searched against a database of proteins. There's a few, but a really well-known one is Uniprot or Swissprot. And so those you can say, okay, I want to look in all human proteins, and this is how I cut them up, so I know what pieces I'm looking for. The software will match the observed mass to charge ratios to pieces of human proteins. And so this takes a while. This is something that I set up usually in the evening. That is the last thing I do before I leave, and I come in the morning and it might be done. So this takes a while depending on the size of your search space. How many proteins are you searching against? If I'm searching in all of the human proteins, again, that takes a second. But yes, the software is what helps us match this list of mass to charge ratios to observed proteins. And to your question earlier about how do you differentiate two proteins, you can tell your software how specific you want to be. Usually I tell it, I want you to only show me proteins that have two peptides that belong only to them. So that helps us differentiate between two or more closely related proteins. And then that's how we sort it, sort of.
Interesting. And so you can tell like how many peptides are in each little, like, just for context for our listeners, what a peptide is it's a basic, correct me if I'm wrong, everyone. It's essentially a chain of amino acids. And so a protein is a big chain of amino acids, and peptides are small chains of amino acids. And when you chop up a protein, you get different peptides. And so, yeah, amino acids are your building blocks. Peptides are like small chains made of the building blocks. Proteins are big chains made of the building blocks. So just adding a little context there. And so just to reiterate, to make sure I understand, you're saying, like, based on these mass to charge ratios that you get in the experiment, you can figure out, you can correlate those mass to charge ratios with different like peptides. And so let's say if I have a mass to charge ratio of, I don't know, like, and maybe this is a poor example. For context, I don't do mass spec.
None of us do, other than Margaret on this call.
Yeah,
we're all like, what's happening?
If I get a mass to charge ratio of, just for very, very like a very simple example, if I get a mass to charge ratio of three masses, or like 3 units of mass to 1 positive charge or something like that, that's different. Like, that's one peptide. And then if it's 6 units of mass to 1 positive charge, for example, that could be another peptide potentially. Is that a
yes? That's a pretty good visual, I guess, a good example.
Okay.
And so, yes, that the software, like I said, is linked up with the database and can tell us what proteins in that list are there.
Yeah.
Yeah.
All right.
Very cool.
So you've
I have a
got.
question.
Oh, go ahead. No. You go ahead.
Okay. Well, I was gonna say, so you're studying the brain, which is super cool. So you have all these proteins that you're lining up through this mass spec and identifying them. What do you do then? How do you gain more information from your overall picture and experiment? What's your next step? If I was designing something like that with a mass spec, what would I want to do as a scientist?
Sure. So having the information from the database saying what proteins are there and how much of them are there, that's a big part of the puzzle. That gives us information about what pathways might be affected. There are other tools and other softwares that will let me sort of parse out what processes might be changing, because in one of my samples, I might have anywhere from a few hundred to a couple thousand proteins. Not all of those are going to be of particular interest to me, based on the processes that we're studying or things like that. But what really happens is I rely very heavily on other sort of subject matter experts. I mentioned earlier that I work with neuropathologists. I also have some collaborations with people in the engineering department where I go to school. And they really help me place that information in context. So if I can say, all right, type 1 collagen is more abundant in my treated samples than my controls, they will help me sort of figure out what that might mean.
And I think, and correct me if I'm wrong, but I think a big benefit of that is, you get all these samples and they're not labeled for you, right? You go in and you're like, you don't go in with any preconceptions. You're just like, I'm going to run the mass spec on these samples, no bias, no background information. You get these results, and then you're able to bring them to people of different disciplines who might have additional insight into that particular area, and then they provide overall context to your results, which you can then use for science. I guess that is science, but like then you do the next step.
No, that is a really good point. So usually what'll happen is I will have picked up my samples from my collaborator, or he's sent them to me or whatever. I will have like an Excel file that has sort of the diagnosis information for the samples. And so I will get a sense of, okay, I have 12 Alzheimer's and 10 control, whatever. But all of these samples have some sort of identifier on them that helps me track which is which. Mm-hmm. And then I don't really look at the Excel file again until I have run the sample, or I've run all of the experiments.
Yeah.
Because I want to do this blinded and in a random order.
And I think it's important, for our listeners to talk about why it's so crucial to do that in a random order. I know it's a huge part of the scientific method, right? That keeps you unbiased. Mm-hmm. You three are the ones actually doing the science, so maybe you could kind of just touch on the importance of not having that bias in samples.
Yeah. There certainly, if you know, in an experiment like this, if you know where, which sample is which, that you might be more likely to come to certain conclusions about the data. Or maybe do different things with your samples before you actually analyze the data. And at least in my opinion. I think one key point that to drive home for our listeners is that, Margaret, correct me if I'm wrong, this kind of research starts off being hypothesis free. So you're not necessarily, or maybe I'm incorrect about this, Margaret, but
no, you're right. These are hypothesis generating experiments, is what we call them.
Okay, and so for just regarding a lot of my research is usually hypothesis driven. So I will say something like, okay, if I think that if I give my cancer cells this drug, then the cancer cells will die. And I could do an experiment where I have two dishes of cancer cells. And one I give a control solution to, maybe just water, and then I give a treatment solution to the other sample, maybe drugs in some water. And I just kind of plot both solutions, one on one dish, one on the other, look to see if cells die or not. And so what I'm trying to do is I'm trying to see if there will or will not actually be a difference between the two groups. And so I'm trying to reject my null hypothesis, which is that there is going to be no difference. And so if this gets a little, I think the logic and philosophy behind this can get a little confusing, but my goal here is to assess whether there is a difference or not. And if I do see that the cancer cells die in the drug treatment group, and I see that they don't die in the control group, I could say that they are different from each other, and I can reject my null hypothesis, which would be that there is going to be no difference. But in this case, we're not doing that. We are looking to see, or we're just trying to see whether there may be differences and what kinds of differences, rather than having a preconceived notion about what specific difference we're going to look for.
Right.
Correct. And with the exception of it being some difference in proteins.
Right,
But we're
but
not
to
saying-
extend the example here a little bit,
Yeah.
sometimes a way that those hypothesis-driven experiments are conceived of is from something like a mass spectrometry experiment, where perhaps Joe has given me untreated and treated cancer cells. I analyze them and I come back and I tell him what proteins are different between them. And then Joe goes to the literature and searches up what proteins, what drugs might interact with those proteins, and then chooses to treat his cancer cells with a drug that interacts with a protein that I found to be different. And so that's sort of the way that it can fit into the context of this.
Yeah.
Joe, your explanation on the cells, treating them with different drugs was a great one. And I think it's, that type of science is maybe what people are most familiar with when you think of science. You know, you want to find the answer to the question. I can imagine myself, you know, if I was in your shoes, Margaret, doing mass spec, getting overwhelmed at the prospect that there really is no question yet. How do you maybe navigate that balance, having to actually find the questions yourselves? You know, do you, like, what are the challenges there? And then maybe what's exciting about it?
Yeah, that's a really, really good question. Because, as you've said, a lot of people are much more used to hearing about experiments like Joe describes, where you have a hypothesis in mind and you go in and you test it. It can be a little bit difficult sort of explaining what I'm doing to, let's say, funding agencies. And so that can be... I've had to practice doing that.
So when you're applying for funding, right, and you have to, listeners, for context, when you apply for funding, you have to write out what you're going to use the funding for. And Margaret, it seems like in your case, you're going to be like, I'm going to look for questions, right? And you got to have to spell that out. And it's not, and just because there's no question doesn't mean that there's no value in that. Like, obviously there have been breakthroughs because of mass spec, and that science needs to be there, that investigation needs to be there. But it can be hard to justify it for people who are fighting, you know, for those funding allocations, for sure.
Right. And so I'm gonna keep harping on this, but I rely so heavily on my collaborators, and the people who help me contextualize it for that.
Yeah. Super, super important. I think that goes kind of to the philosophy that we're trying to drive home for our listeners through really all of our episodes. Like, none of the work that any scientist or really any person does in any one field is sufficient, or like you need other kinds of people doing other kinds of things to help make sense of the information that we obtain in science, in, I don't know, economics, like many other fields as well.
Discoveries don't happen in a vacuum.
Nope.
Right. And
No,
so
they do
it's
not.
great in that I can do the instrumentation sort of work that I love, but place it in sort of any number of contexts.
I have a random fun question. What's the coolest thing that you've used your mass spec for? Do you have like a favorite?
Ooh.
That's a good question, Sarah.
Ooh.
I mean, I am biased towards this project with the arteries, because there's not a whole lot we know about them. Like, I start doing literature searches, and it isn't to say that I come up totally empty-handed, but we don't understand. And so to think about, I could help sort of drive these breakthroughs in this disease that we don't really understand the mechanism of, is is really cool.
Question. Which disease are we talking about specifically here?
We are talking
If you
about
don't mind me asking.
Alzheimer's disease.
Oh, very cool.
And so we have some collaborators who've been studying how arteries change in Alzheimer's.
Yeah.
And so they came to us wanting to understand, okay, what proteins are different.
That's pretty bloody interesting, if I do say so myself.
Pun jail. Joe is being sentenced to pun jail.
No, you appreciate it.
I do.
You appreciate it.
I do. So you
That's what
said
I'm
that,
here for.
they come to you. Why did they come to you, in that particular instance? Like, do you have like a cutting edge technology, like a mass spec that's maybe like more advanced than, what made them pick your lab?
Right. So we have a long history of studying molecules that were of interest to them.
Oh, okay, cool.
I mean, to your question, yes, we do have some pretty cool cutting edge mass spec, but I feel like we'll get there later.
Mm-hmm. I kind of going back to like working with different people in different disciplines. Like in my experience, the upside of working with different people in disciplines is that you get a lot of different like people can as a team you can more effectively explain what you're seeing in the data or things like that, or more effectively come up with solutions to scientific problems or things like that. It seems like the challenge is that at least in my personal experience, the challenge with collaborating with people from different disciplines is sometimes not everyone speaks the same language.
Yes, and that's a really good way to put it. We might, even within my lab, I learned this the other week, or I finally put it together the other week. Mm-hmm. We sometimes use the same word to describe two different things.
Oh, interesting.
And so, yeah, when you start talking cross-discipline, this problem just gets compounded. And so I have to be very careful with my verbiage, and with what words, and I have to be careful to define what I'm saying. I remember a couple months ago in this meeting for the artery project, I used a word that I have not had to define for anyone in my lab, but one of the other professors said, hey, can you back up for a second and explain what you mean? And yeah, so I just have to, again, be very deliberate in what words I use and be careful to define terms that even, that I might use every day, but that they don't hear every day.
Yeah. I guess even though we're all, like, everyone in the conversation in that case, like, are all scientists, it seems like everyone's so highly specialized that sometimes, like, they may not know anything about the other person's research or experience or even the words they use. I think that's why, like, it blows my mind every day that there's just so much information out there that, like, even as scientists, like, sometimes it's like we're just so incredibly different that it's hard to understand each other, which is just intriguing. I guess what are some of the other challenges that you've kind of faced when working with people in different science disciplines, if there are any?
Yeah, so because of sort of the collaborative nature of my work, I have a lot of irons in the fire at any given time. And it's really, really easy to get spread too thin. I have had to be really, really careful in what I say yes to. And last year in particular, I said yes to way too many things. And so sort of at the start of this year, I had to spend some time sort of paring down my list of things that needed to happen, and then I needed to sort of schedule them out so that I could say, okay, if someone came to me, I could check my calendar and say, okay, I have to do this in March in order to stay on track, but can we talk in April?
Professional advocacy is something that is so challenging across, so many disciplines. I struggle with that. I am not a practicing scientist. And just being able to set that boundary and be like, I'm more than happy to help you, but it's going to have to be, if you need my help, it has to be on my time because I've got my stuff that I got to do.
And I am so lucky in that the people around me are supportive of that.
That's awesome. That's so important.
This is huge. And so, it's been a lesson to learn still, but fortunately I have a supportive environment in which to do it.
I guess, why do you like mass spec? Like, why do mass spec over something else? Like, what do you find so interesting about it in general?
Yeah, so I said earlier that you're really just measuring the mass to charge ratio of peptides or whatever it is that you're looking at. And that's, on its face, sounds really simple, but it provides so much information about your system or whatever it is that you're looking at.
Yeah.
And there's a lot of data analysis. I have to sit down and look at either my output from my database, or just sort of the more raw data that I get off of the mass spectrometer.
Yeah.
And it's like a puzzle. I have to spend some time looking at it, figuring out how confident I am in these results. And I have to solve it before I can go back to my collaborators and say, okay, here is what I found.
Mm-hmm.
And sometimes this goes back a little bit to some of the challenges. I think sometimes there's a perception that the mass spectrometer just prints out a picture of what's going on. But it is more like it printed out a 1,000-piece puzzle that I then have to put together. And sometimes, in the worst case scenario, it prints out a 1,000-piece puzzle, but half of the pieces are missing.
And you still need to
go
identify
back to my
the
collaborator
picture
and say, I need more sample.
Mm-hmm.
If you want me to help you, I need more sample.
Yeah.
So, but yeah, it's some of my favorite days have been just sort of poring over the data, and just getting to do that.
What do you look for in the data?
So I look for, it depends on what I'm doing. In sort of the cases that I've been talking about before, I look for proteins that might be implicated in some of the processes that we know to be impacted. From our collaborators, again. That is information that I get from them. And but I also look for proteins that, having done some sort of analysis on it, we're pretty confident are changed, but that hadn't really been uncovered before, because that's an interesting finding too. I do sort of two things. I can look for novel findings, and I can look for sort of confirmation of some things that we know, because that's a nice sanity check. Mm-hmm. That's a nice to say, okay, we understand this system, but there's also this new information about it.
Yeah. And in research too, there's one scientist or one group of scientists or one, institution, say they perform experiment A. That's not the last time experiment A is ever going to be performed. What they find is not the be-all, end-all. So I think that really underscores additional importance of, like you called them, like the sanity checks to, hey, is this still true? Are we still understanding this the correct way? Because there have been times, right, where in science we think we have it figured out, and then boom, like a new discovery, and it's all kind of not all thrown to the to the side, but it really throws a wrench in the understanding.
Sure.
Yeah.
Yeah, but another fun thing about being where I am is I get to use some really cool cutting edge technology. I touched on this very briefly earlier, but one of our instruments has an add-on. We were the first ones to get it in the US.
Wait, what?
There are less than 10. I think we're looking at between 5 and 10 in the whole world.
Oh my gosh.
Wow.
That's really cool. Do you go in and you look at it and you're just like, oh my gosh, I get to use that? That's what I would do. I would literally geek
tomorrow
out over
morning, I'm getting on a phone call with the people who made it because we're having some trouble with it, and they're going to help us fix it.
That's very cool.
And I love that.
So you said it's an add-on. What does it add, I guess, in the case of the mass spec?
Yeah, so the way that mass spec works, the way that you can start to see sort of what order the peptide goes together in, or where there might be changes on the peptide, is by breaking it. And so a lot, the mass spec that we had before the add-on, and it still does this, I should say, like you can still use the base model. It is as if I hit it with a hammer. But that can lose you some information. If you have a modification to the peptide that's maybe a little bit fragile, you hit it with a hammer, you lose information about it. And so what this add-on does is it breaks it in a different way. Now, instead of hitting it with a hammer, I'm coming in with a pair of scissors. And cutting it specifically. And so that is a little bit more delicate, I guess is the word. And so I keep my information about my fragile modifications.
That's really cool.
That is so cool.
And so I think what's so neat about that is that because we were losing information about those modifications, they weren't very well understood. And so now with this new technology, like there's a whole new field of information that is available to us that I get to sort of make more knowable, I guess.
Yeah.
I'm quoting Bill Nye when I say that, but I don't know that he was the one to originate that.
The science guy.
But what I do now and what I want to keep doing is to develop tools so that other scientists can use them and start to get a more complete picture of what's happening in whatever system it is that they're studying.
Joe, this is really throwing me back. And Margaret, I'm sure you've heard the episode too, since you're Sam's girlfriend. The astrophysicist episode that we had with Kenneth, where he talked about how he really focuses on the tools so other scientists can do their work. And I think that is so amazing that we've had two people on this podcast that do that, because I feel like you guys don't get a lot of credit, right? For the tool work that they don't just come out of nowhere. You don't, mass spec, they don't grow on trees, in case
they do not.
In case our listeners didn't know, they don't grow on trees, and you need really people with so much knowledge and background to use them to actually get the insights you want. So I just think that's an area of science that isn't talked about nearly enough. It's just think more nitty-gritty tech, the deep tech of science.
Yeah. From a philosophical perspective, I think a lot of what we understand about the world, in science, is based on what we can actually see and test and measure. And maybe it's hard to test subjective things, but things that we can quantify. Sure, there may be subjective interpretations about what a different amount of protein in one sample versus another means, but we can only do experiments with things that we can actually measure or manipulate
or observe.
And so or observe, yeah. And so like making new tools or making new ways of measuring things like mass spec, like it literally from a philosophical perspective, at least in my opinion, it literally expands what we can perceive about the natural world. And I think that is like the coolest thing ever. And in many ways, like those are like from my field, like of like more molecular biology, like gene editing, like CRISPR, which maybe we'll have an episode about at some point in the future. Like that is like a perfect example of like just a thing that is completely paradigm shifting. And in the same way, I think mass spec doesn't get the same amount of like fame in many ways, but it's probably equally important for understanding, in this case, proteins versus like genes. And so, yeah, I think that's super cool. And this like new add-on that you've mentioned, like I wanna do a collaboration at some point like that. That would be. I mean, like put me on your list of like the 10,000 people who want to work with you so they can understand what's going on in their samples. But one other thing I want to ask you, just to add a little context for our listeners, like how expensive is one of these machines?
Oh my gosh.
On the order of $1 million.
Only $1 million.
I remember this because during my internship in undergrad, our lab moved, just like down the street. But it was still like a whole production. And one of the newest, fanciest mass spec went in a truck by itself because the trucks were insured for $1 million. So
wow,
that is the sort of money that we're talking about.
You handle $1 million a day.
When you put it like that, I can't believe they let me do it. But
don't break it. Sorry, I didn't mean to interrupt.
No,
I
just when you put it that way, it's a little mind-boggling.
Yeah, but also like for our listeners, like I've known Margaret for a while, and of all the people I know, she is the person that I would trust to handle like a piece of equipment that costs $1 million. Like she's like one of the most meticulous and detail oriented and conscientious people I know. And so I think
well, thank you.
Like there's no one that I would trust more to handle that kind of thing. So yeah, just putting that out there. Our mass spec tech is in good hands. Despite me joking about breaking it. But
don't let the intrusive thoughts win. You're like, just want to smash it on the ground. No, I'm Margaret's like, I literally would never do that. Like, why are you making that joke? No,
I've come disturbingly close to dropping some very expensive things. Yeah, but I just need to know better than to try to do complicated things at like 5 o'clock on a Friday.
Yes. Yep.
Because that's the only time that's happened. And like I'm here way too late. Like I just should go home and try again on Monday.
Yeah. Makes a lot of sense. Work-life balance. Yeah. I guess we've touched on a lot. And so what are we talking about? We talked about just some of the really cool, interesting experiments you've done, like hypothesis-driven versus hypothesis-free research, the importance of collaboration, developing new technologies. Like, wow. That's a lot.
Everything you could ever want in a podcast episode, brought to you by us.
Yeah. Margaret, if in the last few minutes here, like, do you have any thoughts or advice for our listeners kind of related to these kinds of questions we've talked about? Like any kind of like final thoughts?
Yeah, just to say that when I started this work, I was super intimidated by it. Like, I had only really heard of mass spec in like my orgo class, and it didn't really make sense to me then. but I had a really wonderful supportive group to teach me, and so finding that sort of mentorship and support has been key. And just don't be afraid to ask questions. Find yourself an environment where you can. If you don't feel like you can, then it's the wrong environment.
So true.
Foster
a place that will foster that growth.
One of the biggest things that made me comfortable in my lab now is I would be in a lab meeting and I would think of a question. I would sort of sit on it for a second, like, okay, maybe they're going to get to it. Maybe this is kind of a silly question, like whatever. And then the senior grad student would raise her hand, and this happened a few times. She would ask either the exact question I was thinking of or something very similar. And that sort of taught me that it was okay to ask sort of whatever question I had in my head. Because I was always going to be kind of hard on myself and think like, oh, I should know this, like I shouldn't need to ask. But seeing that modeled was so important for me. And now as terrifying as it is, being the senior grad student in my lab, I'm trying to model that for like the undergrads and the like junior people in our group.
And I'm sure you do a great job.
Thank you.
Well, thank you so much for joining. It's been so awesome having you on the podcast.
Thank you for having me.
Of course. This is an area that we were definitely super interested in, and doesn't get a lot of coverage. So thought that this would be like a great way to kind of introduce our audience to what you do. So thank you so much for joining, listeners. If you are returning, you know where to find us on social. This episode was certainly pretty derpy at certain sections.
Oh, yeah.
I'm like, dee like.
But that's how we do it. That's how we do it.
Roll.
It's how we show that we are not robots in white coats.
Yes.
But yeah, thank you again so much, Margaret. It's awesome to have you, and maybe we'll have you on again sometime.